כל הארץ, יש אחד מה...
זה, חשבתי שיש כל הארץ. פה,
לא כמו ביבי, ולא רוצה לי. שלא רוצה.
יש כאן הרבה שרצות.
זה לא גומר בוקר, כאילו, אתה הלכנו פרצה.
פחות מהשרץ. So it's a pleasure to introduce Dr. Aviv. Guy did his PhD in neuroscience with Illuminating. His postdoc at the Mass Bank in Florida with, sorry, I don't know. Okay. And he started his lab at Aviv University in 2020. His lab uses an integrated approach incorporating optical biosensors, gene editing, in vitro and in vivo imaging to unravel the basic operating principles of neuronal signaling in plasticity and development. And we're looking forward to your talk. Thanks for coming.
Thank you so much. It's okay? Too dark? Good? Okay. All right, so thank you so much for the kind invitation and the introduction. It's a great pleasure to be here, this beautiful building. So today I'd like to show you our work, and I'll just start off with introducing the main question that's driving our interest in the lab, and we're trying to understand how do protein signaling networks regulate dynamic brain function. And as you all know, the nervous system operates on a vast time scale, right? From millisecond to second to structural dynamics that operates in seconds to hours, and gene regulation that occurs in minutes to hours and months and even our lifetime. And all these functional and structural phenomena have intricate machineries of protein signaling inside neurons that regulate these cascades. And this is what we're trying to understand. How does this interplay take place? And mainly looking at dynamics of protein signaling and how this has to do with dynamic neuronal function and this interaction between them, which is not something we often think about and study. So traditionally, as you all know, this has been studied tremendously in the nervous system, and the conventional way I would say that people would like to address this question is by looking at, if we look at the behavior of a mouse and neuronal activity that represents something to do with the behavior, and we have this other axis of protein signaling, and then most commonly people would just take a hammer and smash their favorite gene or protein, knock it out, and see what happens in regards to neuronal activity and behavior, and there we can, point the figure to some correlation and maybe causation of what goes on. And this is a very common approach. It's been incredibly useful in identifying key genes and proteins. But I'd like to argue that, although in some cases it definitely leads us to the right direction, it doesn't really allow us to study protein signaling dynamics. And some of these aspects of the regulation between protein signaling and neuronal activity are lost in this process of loss of function. So this is my small manifesto of why we should study dynamic protein signaling. So as you all know, protein signaling is dynamic. I don't need to prove that we all know this exists, right? It's activity dependent, depends on neuronal activity, and it's compartmentalized, especially in neurons that have huge processes of dendrites and axons, and these protein signaling occurs differently in the cell body and dendrites, so I'll show you later on. And as such, the mechanism of neuronal development and plasticity, as I said, were mainly studied using global perturbation, but in order to truly understand, or to gain a deeper understanding of how circuits form and function, I would say it's critical to have a quantitative approach of monitoring protein signaling both in spatial and temporal resolution. And we hope that this will allow us to monitor the dynamic interplay, and we think this has a deep relevance to disease because all of the targets we study in the lab that I highlighted here, we know what happens when they're perturbed or knocked out, such as autophagy, PTEN that I'll discuss later on, MECP2, CREB. These are all proteins and genes that we know from animal work and human studies that when we have mutations in them or loss of function, perturbation, terrible and neurodevelopmental and cognitive disorders occur, and we think that another aspect of studying this process is understanding first what is the physiological role of protein signaling to gauge it as critical elements, and then go and maybe study how this physiological process is perturbed in pathophysiology. Okay, so how do we do this? So basically in my lab, we develop biosensors. As I'll show you later on, we usually use FRET-based biosensors. I'll introduce in a second. And so we build ways to look at protein signaling dynamics in cells, and then we can take it all the way and put this in introduce this in the brain of mouse and study protein signaling dynamics in the intact brain and tie it to neuronal function using calcium imaging in this example. And we think this is an approach that allows us to really start to gaze an appreciation of this interplay within spatial and temporal resolution. So today I'd like to share two stories. One is what I worked on during my postdoc using a tool to study CREB dynamics and how it helped us gain some information of how CREB works in the nervous system and how it's regulated by activity-dependent and the sensory experience. The second is a paper we just, which I'm in revision now, and this is a biosensor for PTEN, which I'll explain on, and then maybe I'll have some time to touch upon some autophagy work we're working on. Okay, so CREB, probably infamously known for anyone, it's been maybe one of the most well-studied gene in the process of synaptic plasticity and memory. And right, if I'm trying to summarize decades of work and really narrow down of what we think about CREB is that people usually look at this process as the calcium coming in from synapses, right? It enters the cell and then somehow it transfers to the cell body, and then CREB as an activity-dependent transcription factor that many of those exist is phosphorylated, activated, and activates a cascade of genes. And from a long line of work in many different species, from Aplysia to Drosophila in mice, CREB has been pinpointed as this central junction conveying short-term to long-term information storage. And we know all this mainly from loss of function studies and some gain of function studies, because when we perturb CREB by genetic means, in most cases we see perturbation of long-term plasticity and memory. So this is the prevailing view, but when I started thinking about this question, I was kind of fascinated with such a central signaling cascade, and we really have no idea of how this is dynamically regulated, right? All the measurements that have been done are statically in one time point using immunohistochemistry or biochemistry, and they can only reveal to us globally if CREB is activated or not. And they don't allow us to do this in single cell resolution and link it to activity in real time. So this is what I set out to do, and in order to create a biosensor for CREB, what we do is use FRET in the lab. So I think FRET is probably, most people know how it occurs. It's basically the transfer of energy between one fluorescent protein and the other, given that they are in close proximity to one another. And cell biologists have been using FRET for many, many years now. But when we try to take a FRET biosensor and implement this in a complex biological system such as the brain, we're tackled with many, many problems, technical problems and conceptual problems, right? So most people that use FRET use just fluorescent intensity to measure this, and this is inherently noisy, as everyone who has ever done imaging knows. We have light scattering in the brain, and unfortunately, unlike GCAM, the signal to noise of a typical Fred biosensor is very small, the range of 10 to 20%. So it's really not that hard to think of how we could not distinguish this signal in an intact brain, and it's hard to use. And as I said, small dynamic range, and obviously if we want to do this longitudinally in the same animal, we're faced with fluctuation in expression levels and whatnot. So it's basically almost, I would say, impossible to use Fred biosensors using fluorescence intensity over long time periods. During my postdoc, I went to the lab of Ryohei Yasuda, who was one of the pioneers of the use of fluorescence lifetime imaging as a way to measure FRET. So basically, fluorescence lifetime is the time between an excitation of a fluorophore and the emission of a photon, and we can actually measure this process. And there's many advantages using fluorescence lifetime as a mean to assess FRET. It's basically almost completely independent of the concentration of a fluorescent protein, so we don't have to rely on how much light or how much expression we have on a cell-to-cell basis or a daily basis. It allows us almost a biophysical absolute quantification of binding, and it's relatively insensitive to all kinds of issues with imaging and intact brain, such as wavelength-dependent scattering with two-photon imaging. So it's ideal for measurements in the brain. And as you'll see, it's relatively straightforward, so we don't need two fluorescent proteins to worry about. We only need one, the donor, because this gives us all the information we need. If anything, if you have any question, you can stop me, whatever. And then we can use other channels to look at GCAMP or neural activity or other sensors. And so the way I set out to do this was basically develop a biosensor for Krebs from decades of work. I already have a lot of it. We already have a lot of information of how this complex of Krebs is situated inside these key genes in the DNA. It's a dimer that binds other obligatory proteins, but the critical association here is when Krebs gets phosphorylated, and there's a specific serine, which is the bottleneck or the critical serine for activation. We knew that this specific protein called Krebs binding protein, CBP, binds to it. So I designed this biosensor based on this interaction, knowing that if we see increase in FRET and increase in binding, we can use this as a proxy of Krebs activation dynamically. And so obviously this works, otherwise I wouldn't be talking about this. So when we tested this in cells, we can, using pharmacology, activate Krebs using forskolin, and then you can see this warmer color is indicating higher binding or increase in Krebs activity, and this point mutation of this specific serine completely blocks this binding and this association. So we have a way now to measure Krebs in living cells over time that we lacked before. And so, making a very long story short of how to transfer a sensor from cells to in vivo, which takes a long time of optimization, but in this case, it's only one slide, so we're using AVs to label cells in the somatosensory cortex in this case, and we have one AV for a donor, Krebs labeled with GFP, and the acceptor, which binds to it when it's phosphorylated. And we have these relatively large field of view that allows us to study many, many different cells at the same time. And so we started looking to what we see in vivo, and we saw a surprising level of variability among single cells. So this is just a snapshot of an anesthetized animal, and each color code represents different level of Krebs activity. And you can see from this looked a little surprising because we didn't have any quantitative ways to measure differences between different cells. All we had was a way to say Krebs is activated or not activated. So we did some control experiment, and we were we argued that this variability, at least in large part, is due to biological variability, since we use this dead sensor or non-frustratable sensor as a control, and we see this reduced variability and lower binding. So this tool gives us the handle of measurements of Krebs activity in living animals. So we did some some experiment to show that this really, we can use this to show some biology. So in this case, I'm just showing you one example of what we did. We look at the same cells from the same animal. In this case, you can see the same window of a bunch of nuclei from our Krebs sensor, and we can track the same cells over a period of 3 days when the animal is basically doing nothing, sitting in its home cage. And when we put it in an amusement park and show it an enriched environment, we can see this dramatic increase in Krebs activity across pretty much all the cells. So this and other experiments we performed allowed us to validate this tool and show us that you can indeed track Krebs activity over time in the same cells. And we see some correlation with sensory enrichment. And so, but this gave us, this was very interesting, but it really didn't give us a clear idea of the question I posed to you earlier, which is what is the link between neuronal activity and changes in Krebs activity? Because all I showed you was that we can see this increase in Krebs activity, but we don't know anything about how these neurons behave in terms of function. Yeah?
How do you know it's not a motor effect?
It could be. This is a really global phenomena, yeah. It could be also a motor effect, right? So this is the somatosensory cortex in animals, yeah. It could be not precisely linked to its motor function. We did do other experiments in the motor cortex specifically where we trained animals in rotorod, and we see the correlation between learning basically, or how well animals increase their performance and increasing Krebs. And so what we really were geared at doing now, and we felt that we needed to examine this interaction between neural activity, or calcium in this case, because that's the second messenger that activates Krebs eventually, and how is this interaction occurs in vivo, and how is it linked to sensory experience. So to do this, as I mentioned, or I don't know if you noticed, but we had two fluorescent proteins in this case, one green, one red, so we really didn't have any optical way of looking at activity using calcium-dependent probe. So I switched geared for a short period, and we decided to optimize red fluorescent protein that will allow us to track Krebs activity or any biosensor activity using a FLIM pair and pair them with GCAMP in this case, and that's what we did. Well, these are called CyRFP, and this was a collaboration with Michael Lynn's lab. And basically, the nice thing about these proteins is that they're excited in the same wavelength, so you don't need to tune your 2-photon laser to a different wavelength. But they have almost completely separated emission, and thankfully for us, they're very bright, and we can use them for lifetime imaging. And we did some careful measurements in slices in this case, and we wanted to, in a way, repeat experiments that people have done using immunostaining and see if we can see these two processes simultaneously, calcium and Krebs at the same time. So in this case, cells were labeled with a red shifted Krebs sensor and a GCAMP as a probe of calcium. And this is with extracellular stimulation. As you can see here, we can see this quick, unsurprising burst of calcium, because this is us stimulating the cell, and this slow buildup of Krebs phosphorylation within the time course of tens of seconds. This matches previous measurements done with immunostaining. So this experiment allowed us to prove to ourselves that we can indeed measure this correlation in real time. And then what we really wanted to do with this tool is measure this correlation, or this interaction in vivo. And so this is what we set up to do, this time labeling animals with our red sensor and GCAMP. And as you can see here, we can now track, so the magenta here just represents Krebs positive nuclei, and we can track neuronal activity and lifetime at the same cells, and we don't see any optical perturbation between the two channels. So we think we can separate these two channels independently. Okay, so what we used the visual cortex, and what we geared up to do was something that we thought was a relatively simple experiment to start to understand this interaction. We wanted to take the animals, impose them to dark rearings. This has been a heavily used and well-studied system, which, there's a lot of papers that try to understand what happens in dark reading, but there's some even in adult animals, and this is where we did our experiment, we see this, people see an increase in plasticity in the adult visual cortex. And we also knew that dark reading basically alters a lot of activity-dependent genes. So we thought this was a good starting ground to start to understand this interaction between calcium and neuronal activity and an activity-dependent transcription factor that should activate genes. And so this is what we did. This is, I would say, almost a naive experiment, but the result, I think, was surprising. So what we did was took animals, either naive animals or dark-haired animals, and basically put them for 30 minutes under the scope, awake animals, and showed them the same visual stimuli. It's a naive movie that plays again and again over a minute period. And we tracked Krebs activity in this 30-minute period. As you can see here, I don't know if it's very apparent, but we see some increase in Krebs activity, not very robust. And then if we look at the same cells a day later, they're back in their basal state, and we don't see any evidence of this visual experience. And this is the average of all cells. You can see there are traces for single cell on the average, which is colored. We can see this slow accumulation of proliferation over 30 minutes, and if we measure again after 24 hours, we see the signal back. What we saw after dark green was really different and surprising. We saw this much greater accumulation of Krebs signal. This signal was augmented and much higher after 30 minutes, and it remained elevated for at least 24 hours and even 2 days if the animals were kept in the dark. So,
can you remind us which region is it?
Mice.
Which region?
Visual cortex. Sorry. Yeah.
And what is the sensory stimulus here?
So this is just a naive movie that the mouse is watching. It's like a GoPro from a dog walking on the grass. So it's just some visual stimuli that we didn't initially pay a lot of attention.
And it's shown for how long?
It's a movie of 1 minute, and we show it again and again and again for 30 times.
For 24 hours?
No, for 30 minutes, I'm sorry. So we show this visual stimulation for 30 minutes, then we take the animals back and image it again after 1 day. And so this left us a little puzzled why we see this huge difference in Krebs activity from the same visual experience, basically. So this movie was played here in dark reared and naive, and we see this greater response in Krebs in this group of animals. So we thought the trivial answer to this was just more neural activity, right? So more calcium coming into cell, maybe the animals that were in dark rearing now respond to visual stimuli with increased neural activity. And so we had GCAMP in the cell, and we analyzed in the same group of cells how calcium accumulated over these 30 minutes. And I'm just showing you a representative trace of a cell that responds. So every line here is the visual stimuli that goes on for 39 seconds, and you can see here increase in calcium. So this cell responded to certain elements within the visual stimulus, and if we now plot this cell throughout the 30 times it shows us, we see this relationship between increasing Krebs activity and the summation of calcium throughout all these trials. And so this builds us a relationship that shows us how much calcium we need, or how much calcium is going into the cell, and how much this increases Krebs activity. This is an animal that we're dark reared. What we see, and this is a representative cell from a naive animal, where it looks very different in terms of the relationship. We see a much lessened accumulation of Krebs activity to basically similar increases in calcium. And then when we average all the cells and build this plot for dark-reared animal, naive animals, what we saw that basically we didn't see any difference in terms of how much calcium went into the cell. So if you use GCAMP and did this experiment, I doubt that you will see any difference between dark-reared animals and and control animals. Well, we did see that crib differentially responds to the same amount of calcium with increased level of activity. And as I said, this persisted for at least 24 hours after the reporting, which in the naive case dropped down to 0. And so we can conclude that what we see is that dark green increases the sensitivity and duration of crib activity to basically similar calcium levels. So just to summarize this part, I've shown you that we've developed an approach to simultaneously measure calcium dynamics and crib activity in the awake-behaving mouse brain, and we show that sensory experience in this case, dark green, can increase the magnitude and duration of crib and its sensitivity to similar calcium level, which we found quite surprising, and we're still actively working on how this can happen in terms of mechanism. So I'll just move on to switch gears to the other probe that I mentioned, PTEN. So as you all know, during early development, neuronal circuits have to maintain this balance between excitatory and inhibitory drive, and this is a really sensitive period for the development of this balance that stems from both neuronal activity and proteins and genes that are critical for this regulation. And I was struck by this paper that really drove my attention to PTEN as a mediator of this process, based from the lab of Oscar Marin, published a few years ago, where they showed that during early postnatal development in the mouse, we can see this changes in the number of neurons, basically excitatory and inhibitory neurons, until we maintain this stable population of neurons, excitatory neurons versus inhibitory neurons. And you see the switch in early postnatal development. And so they were trying to explore this mechanism, and they found that indeed neuronal activity was one driving force to reduce the number of inhibitory cells. But in terms of gene or mechanisms, they claim that PTEN, a phosphatase, is the mechanism involved in this, and they had some biochemical data that showed that PTEN might be operating differently in this first postnatal week. And
could you explain the y-axis? What's neuronal variation?
I think it's in correlation to the number of neurons, basically. So what they're claiming that this is basically a numbers game, that what they were looking at, not function. There were no functions. And so what I was struck by is this really important mechanism involving PTEN, but more by the way that I felt that this requires a method to visualize this. It was in text vectors, and you can kind of speculate based on this that this is really not trivial experiment. There's a lot of noise associated with grinding whole tissue and looking at activity-dependent signaling. So I set up to explore this question, and we focused on PTEN, and there's really a large amount of literature, again, involving loss of function data from PTEN, which is, I would say, mostly famous from cancer field because it's a tumor suppressor gene, maybe one of the most classical ones found. Roughly around 40% of human cancer tissue contain PTEN mutation, and you can understand why, because basically PTEN is a phosphatase that inhibits cellular growth and proliferation in all tissues, not just the brain. And so when we remove this inhibition, basically cells become hyperproliferative and start increasing their size. In the brain, similar things happen. So people have used this model to show that loss of function of PTEN leads to a wide range of pathophysiology, basically aberrant soma size, microcephaly, a lot of pruning defects, too many spines, as you can see. Here, it's really a dramatic effect. And some of the ASD models in mice were developed based on PTEN perturbation, because we know from human clinical data that there's a tight correlation between mutation in PTEN and the occurrence of ASD, mainly very severe ASD forms in people. And so, on the other hand, if we increase P10 function, we see exactly the opposite. Basically, reduced cell size, microcephaly, enhanced long-term depression, and enhanced social interaction. So this seems to be a critical signaling protein that maintains this balance of structure and function of cells, particularly during development. And so the first MD PhD student that joined my lab, we set out to build a biosensor to look at P10 activity. And so nature already did the hard work for us, because P10 is one of those proteins that changes its conformation when it gets activated. So we can take P10, put two fluorescent protein on both edges of it, and basically, as you can see here, when it gets activated, it opens up, and we can see this in living cells and see an increase in its activity. And this is quantified here with pharmacology. The timescale is really slow, but this is mainly due to the fact that we don't have direct pharmacology to go after P10. And so this looked like a really useful tool, and we set out to establish it. This is indeed useful in a variety of experimental systems. But we were troubled with this that I told you just a second ago, because our biosensor used P10 overexpression, which I just said is really bad for cells. So we took a long time and tried to optimize this biosensor. What I mean by optimizing, we screened for many, many different mutations that I'm not showing here, which allowed us to basically perturb the intracellular signaling of our sensor, but still maintain its dynamic range and its conformational flexibility. And we found such mutation. I'm just showing you one of the things we did to prove that this sensor is basically inactive in the brain. So here, what we did is I would say, to probe how cells respond to P10 when we knocked out endogenous P10. So you can see an example here of a control cortical cell. And this is the cell after we knocked out P10. This is the same image size. So huge increase in P10, as I mentioned before. Dendrites are bigger. It's a very clear pathology that we can see. And so this is P10 knockout. Then when we reintroduce back our original sensor, which contains enzymatic activity, we can completely rescue this knockout, meaning that this sensor is active. And then when we use our non-functional sensor that we think has greatly minimized activity, the cells are still elevated due to the P10 knockout. So this and other things that I'm happy to go in depth afterwards. We think that this is an optimized biosensor with which we see similar dynamic range of changes in thread, but really no structural and functional phenotype that we can see when we overexpress this in cells. We also made some interesting transgenic animal using C. elegans. So this was a collaboration with a lab next door that wanted to study P10 in the worm, and then we made a really cool use of the biosensor. We basically made a transgenic worm that had P10 expressed in all the neurons, and you know, P10 is evolutionarily conserved in worms, and so it has been linked to longevity and proteostasis in the worm in the same signaling pathway that involves IGF-1 receptor. So it was interesting. They used a small animal that only lives for about 3 weeks, and we could even do experiment tracking all different developmental ages of the worm, and you can see this increase in P10 activity in control worms that is interesting to look at, since it seems to be correlated with the development. And then when we manipulated the IGF-1 receptor analog of the worm, DUF2, which prolongs the life of the worm, we see this flattening of P10 activity and its saturation throughout developmental phases. So we think this is a really useful biosensor for a variety of experimental system, and also, you know, yeah.
Right. So you're basically putting in your own P10 and seeing if it's activated. Right, that's the idea. So are you seeing the amount of P10 that other P10s in the cell, or are you just seeing how much yours is activated?
No, what we're seeing is basically how it opens and closes on itself, right? This is the biosensor. This is on top of endogenous PTEN. We're not knocking out endogenous PTEN, but we think this is basically a proxy of PTEN activity, not the amount of PTEN, but the activity itself. Since PTEN activity is correlated, there's mechanism that restricted when it's closed, it's restricted conformation to a closed conformation. When it opens up, it's active basically. And if you open it up, you basically have a considered active version. So what we're looking at is a proxy of PTEN activation, activity state, and not its amount.
Right, because what you're actually sensing is whatever it is that's opening it, right? That's
yes, exactly. The upstream, yeah, exactly. You're correct. Yeah. Okay, so we think it's highly useful for a variety of animal models, and as I've said, PTEN is very well studied and an important target in cancer, and we think this probe can be useful in that field as well, and we actually have an active collaboration to try to use this for high throughput drug discoveries for PTEN-related pharmacology. Okay, and so what we focused on is measuring PTEN in the mouse brain, as I mentioned, and we can, again, use the neural electroporation, insert this plasmid that expresses the PTEN sensor, and then use force and lifetime to quantify its activity. And you can see we can already, just on very coarse measurements, see that we see different activity or different compactness of PTEN that originates in the cell body and in processes in dendrites. So we see a consistent reduction in PTEN activity in dendrites and synapses as compared to the cell body. And this is mouse somatosensory cortex, SR23. And so again, we set up to see if we can perturb any of these signaling pathways which regulate PTEN either upstream or downstream, and see if we can use this sensor to read out what goes on with PTEN activity. So this is a control cell that we can see a cell body and a process, and when we knock out IGF-1 receptor, which is upstream of PTEN, we can see this elevated levels of PTEN activity in the cell body. In the processes, it's fairly interesting. We see what looks like a difference in dendritic activity as compared to the spine. And another example here is a TSC2 knockout, another autism risk gene. And we see this, as you can probably see, this also increases the cell body size and also increases PTEN activity. But we see an opposite trend on what goes on here in terms of spines. We see a decrease in PTEN activity in the synapses. And we think this subcellular and even synaptic compartmentalization can start to teach us something about the spatial and temporal dynamics of PTEN activities that was basically impossible to describe before. And so, as I mentioned, we were trying, again, to see how this activation of PTEN is linked to neuronal activity. So to do this, again, we red-shifted our sensors with our favorite red fluorescent protein donor and acceptor, and this allows us to simultaneously express GCAM and a red version of PTEN and measure neuronal activity and PTEN activity in the same cells. And we found a significant correlation, negative correlation between overall spontaneous activity and PTEN activity in naive mice, which we thought was interesting since, again, loss of function of PTEN has been linked to autism, but also to epilepsy. So this makes sense in a way. And again, we can use this tool now to interrogate both PTEN function and neuronal activity in the same cells. But I think what I was getting at from my introduction was something that caught our attention most of all. Can we really look at PTEN activity in two different cell types, right? Something that was very difficult to do with biochemistry, and probe cell-specific function. And this is what we set out to do. So in this experiment, we took two different viral variants of PTEN, one that is green and one that is red, and using transgenic animals with PV3, we can label them with a green variant and look at excitatory cells with the other variant. And as you can see in this image and this close-up here, we can basically have in the same mouse both variants labeled, and we can separate them well with versus lifetime due to their different properties. And we did what we thought was an initial test to test if indeed excitatory inhibitory cells have different PTEN signaling, which was a naive experiment. What we did was took animals during their first or second postnatal week in the somatosensory cortex and did unilateral whisker trimming to basically block sensory experience in one side. And what we see is actually interesting. We see that this inhibitory variant of PTEN after whisker trimming show increased PTEN activity, and actually the opposite happened in excitatory cells, so decrease in PTEN activity. And we think this is just the initial step, but we think this is a first direct demonstration of a cell-specific PTEN dynamics, which was really hard to look at before. And this is just a little ongoing work that we're still puzzled about, so if anyone has any ideas, be happy to hear. So as I mentioned, PTEN is heavily involved in shaping this early postnatal E to I balance. So what we did was just track the activity of PTEN in really young mouse ages from P8 to P10, P14, and P45. And what we see is something that we think is an interesting phenomenon. We see this, what we refer to now as a critical period, or an increase in PTEN activity in a very precise cortical development age. We're still not sure if this is experience dependent or not, or this is genetically hardwired, but we think this is really interesting phenomena that we're looking at now in terms of function, because our collaborators in Denver actually did structural and physiological measurements, and we know that if we perturb PTEN before P8 and after P14, basically we see very different phenomena. Basically, if we do this early on, we see the structural deficits that I've showed you earlier, but if we perturb the gene after P14, we see very little evidence of something going on. So we think this indicates a kind of a critical period for PTEN signaling activity that I think this kind of approach is useful to discover. And of course, this has relevance to autism spectrum disorder, because we know it also is heavily associated with precise developmental changes. Okay, so just to summarize this part, so I've shown you that we were able to engineer and optimize a PTEN flip sensor that we showed to minimally interfere with endogenous PTEN signaling. And we can use this in various in vivo models with precise spatial and temporal resolution. We also developed a redshifted version that allows us to simultaneously monitor other biosensors. And we think we're demonstrating cell type specific PTEN signaling for the first time. And our hope is that using this approach, we and others can start to illuminate and look at PTEN signaling dynamics in intact biological systems, and especially during development. So I'll just quickly go over, just a couple of minutes, go over this last project in our lab that I'm really excited about, involving neuronal autophagy. So autophagy is kind of the recycling factor of the cells, if one wants to make analogies. So basically this is a process where proteins and lipids get recycled in the cell and sent to the lysosome, and really we know a lot about autophagy. In neurons, we know that in the brain, unlike peripheral tissues, autophagy always works in height flux from the moment the neuron is born until it dies, basically. And it's highly compartmentalized. We know from many decades that this process in the soma and axons and dendrites operates in a different time scale and molecular identity. And it's really a dynamic process. And my attention was caught to this due to the link between autophagy and neurodegeneration. So if we look late on in neurodegenerative models and human brain, we know that due to accumulation of toxic or misfolded protein species, we see this gradual increase in autophagy, or there's some debate if it's an increase in autophagy or just a failure in the autophagy process itself. So this seems to be a really important process regulating structural plasticity or perseverance, and is clinically relevant. And so there's been a lot of methods developed to study autophagy, maybe more methods than any other cellular process. But I would say that most of them lack a clear quantitative way to track this in a dynamic plane that we were geared on doing. So what Yossi and my lab did was basically take a previous approach. So this is Fred pair that you can see here. This is an unusual blue fluorescent protein, and it's completely and utterly pH insensitive, which is really unusual for fluorescent protein. And you can see this, we're tracking lifetime, and it's really incredibly resilient. But when we pair it with a regular XFP that is pH dependent, we get this pH intracellular monitor, basically, that allows us to now quantitatively assess pH levels inside cells. And what Yossi did was attach an autophagic cargo LC3 that is traditionally used in these type of biosensors. And this allows us to now look at autophagic vacuum when we induce autophagy in cells and quantify it. And this is still ongoing work, but I just wanted to share some of it because it's really exciting where we can take this. So now we can, this worked extremely well for in vivo imaging. We can take a structural marker and this autophagy marker and label cortical cells. And you can basically see, this is in vivo in an anesthetized animal, you can see an exquisite resolution, basically different autophagic puncta on every cortical cell, and even in processes next to synapses or on the dendritic shaft and some inside the spine. And so, and I think the big difference is that we can quantify it, but we can also see some incredible dynamics in vivo. So this is, again, taken in a live mouse, and you can see this autophagic puncta moving around inside the dendrite on a second time scale and accumulated inside the spine. And again, we can quantify and see how mature this vacua is. And what we're doing now is trying to do in vivo imaging and then trying to do some really, I'd say, almost seems like trivial experiment, but I think some that we still don't have a clear answer for, such as how does food deprivation for a short period of time changes autophagy in the brain. So in the muscle, we know very well that there's increases, but autophagy levels in the brain are already very high. So I think we yet to have a clear, definitive answer to this. So we have some preliminary data that we're collecting now, and we're also doing a bunch of genetic manipulation to see how autophagy changes in disease state. Yeah, and then mainly it's fun to watch because there's a lot of these dendrites moving around, and yeah, we're trying to understand how they shape structural stability or failure. So yeah, this is an axon. This is so that's it. Just want to acknowledge my students that did the work, and my collaborators, and my funding sources. And thank you.
Thank you, Christy. Back to the first Krebs story. Do you have some leads? And so if it's not calcium signaling, should be some reprogramming, maybe of CDP or some other Krebs?
Yeah. So I mean, we think that at least one mechanism responsible for this was this shuttle. So there was from the lab of Dictian, they identified this kinase that they think shuttle calcium signals from spines to nucleus. There's a lot of these proteins that shuttle between synapses and the nucleus that they study, but this one in particular seems to be important for Krebs signaling. So we do have some preliminary data to see that some of this differential regulation is at least blocked by ablation of this. So this is kind of interesting, but we're not, in most cases when you try to mess with activity-dependent signaling, you either mess with activity, which changes a lot of things, or the downstream signaling. So in this case, we're just knocking out the shuttle and not changing calcium coming into the spine, and we see the changes in Krebs signaling, which we think is that's probably one mechanism. We've tried a lot of other things like adenosine, adrenaline, stuff like that we didn't see any evidence that are the cause of these type of differential activation rather than calcium. Yeah.
Early on in the beginning of your lecture, you mentioned involvement of crabbing in molecular memory. Can you say more words about it?
Yeah, I think, so as I mentioned, a lot of people showed that basically loss of function of Krebs is important for long-lasting plasticity. And what people meant there is just this not short-term plasticity in terms of minutes or hours or something that happens over a period of day. And I think it's possible that the mechanism that we're showing, we have some good evidence to show that this might be the reason why, because what we're seeing is this a matching that always, to me this was always something that I didn't really buy, so to speak, in the activity-dependent world, because if you look at a culture and you say this transcription factor is activity-dependent, neurons are always active. There must be some mechanism to restrict this activity-dependent neuron calcium, and I think there's very little mechanism that describes as it kind of tilt the balance, and because otherwise it's a positive feedback loop that never ends, right? You need something that. So I think this at least partly starts to lay the ground to see how this experience changes this interaction. So Krebs is responding to calcium, but I think there's other cues that could change or modulate this. So this is my 2 cents of how Krebs might be involved in memory. Yeah.
Relatedly, it looks like at the 24-hour time point, the Krebs much better distinguish between the active and the inactive cells.
Yeah.
Do you think there's any regulation of phosphatases that are leading to the prolonged phosphorylation state, and maybe building sensors up?
So that's a great question, and I think this is kind of an enigma in the field, because I think there's activity-dependent genes that have also shown this long-lasting regulation. Probably FOS and others after all kinds of in vivo perturbations. So I think this might be involved in these long-lasting generation, but I should also be careful and say that we're monitoring phosphorylation of Krebs, right? So this is, as every probe has advantages and disadvantages. I think the advantage of this is the on-kinetics. The off-kinetics is, as you say, probably biased by phosphatases, and it's still unclear to us how much, what does this mean, basically, that Krebs remains on for, it puzzled us also, yeah.
So the story that I've heard is that, so in the early phase of plasticity, you bring the receptors into the spine, and then the late phase, which is protein synthesis, which I presume includes Krebs. So at least part of that story has to do with structural proteins in the spine to stabilize the additional receptors.
Right.
So do you believe that story, or do you think that it's something additional?
It's an interesting question. I don't know if anyone has definitive answers there. Probably not me for sure. But yes, if you follow that theme, that maybe this long-lasting activation of Krebs is involved in genes that go back to the activated spine or synapses and increase their structural stability or changes their properties, right? This is the theme. We actually had some initial project where we tried to monitor both processes at the same time, and unfortunately we failed but this is an interesting idea, how to kind of track synaptic protein and Krebs at the same time and see if this is indeed, this signal leads back to the spine. Yeah, we haven't done this. Yeah.
Very nice. So in the darker animal, how long does the signal stay after 24 hours?
So we've measured, so if you keep them in the dark at least 2 days, if you bring them back to normal night, it dies out within a few hours. So it's definitely experience dependent, meaning that you need this prolonged darkness to maintain this elevated level.
But if you keep them in the dark?
Yeah.
So you said if you turn them back to light, it was a signal.
Yeah,
but
yeah.
If you keep them in the dark?
Yeah, at least for 2 days. That's as long as we measured it, yeah. Yeah.
And on top of that, if you now use another experiment, do you see other cells turning on?
We haven't done this. I don't know.
But I always find it strange, the specificity, right? I mean, single cell specificity as well as the specificity of the crab in terms of the entire genome, because it looks like it's everywhere.
Right.
Also with the P10, it's like your substrate is everywhere.
Yeah, I mean, I think we like targets that are ubiquitous and are important. I mean, yes. I mean, if you knock them out, you definitely see, strong response in any cell type you choose. So I think, yeah. I mean, I don't have a good answer to this, but yeah, it does seem to be a really robust and ubiquitous. Yeah.
Given that the dark yards show the effect only if you bring them back to the dark, do you think you could do the opposite? Take the naive, put them in the dark for a while, and then...
So we've played around with this. It definitely seems like, so these are adult animals, and usually in younger animals, people tend to do a shorter period of darkering that is enough to change the circuit, but in adult animals, you need something in the order of a week to actually, at least in terms of function, right? So we've done some initial experiments, and we did see a smaller magnet, and when we didn't do the experiment you said, but we did keep animals in dirt for 3 days, and we saw a similar phenomena, but the persistence was slightly lower. Yeah. All right. Thank you.
Okay.
Thank
you
you.
very much.
ממש מגניב.
כן, תמיד גונבים לי את זה, אז אני רגיש.
מישהו עשה כאילו בחושך הזה, לדעת שינוי באפיגנטי? אז כאילו מה שמציע שהוא עושה לך איזשהו שינוי בחלבונים קיימים, איזשהו כינס, ואז הקרב קופץ.
כאילו, עשו דברים, כאילו זה נראה שהכל משתנה, כאילו זה נראה לי יותר מדי זמן, יש עבודה ממייקל גרינברג שפשוט נראה שכל סוגי התאים מגיבים אחרי, או כאילו גם...
לא, כאילו בבייסליין, לפני הפעילות. כאילו זה מציע שמראש יש לך לדוגמה יותר רדיאציה קלאס, יותר חיתוך, וזה מגביר לך את הסיגנל של הקלציום.
מהמעט שעשינו, בלי אור, כלומר, הסטריגיה שאנחנו ראינו זה אור אחרי החושך, כלומר, אם החושך עצמו didn't do anything, כאילו אנחנו לא ראינו...
לא, בגלל חשבתי באפיגנט.
לא הסתכלנו, אבל אנחנו, כאילו, חייל בחושך, בלי שחשפנו אותם לאור, לא ראינו שום שינוי בזלי, בוא נגיד ככה.
הסרטון בסוף, כמה זמן זה?
עשרות שניות.
וברזולוציה של מה? צריך ננו סקנד?
לא, אנחנו עושים אקומולציה של סיגנל בעצם, אנחנו מודדים בסוף בננו סקנד, אבל זה accumulated, כי אנחנו צריכים עשרות אלפי פוטונים פר פיקסל כדי להשיג עקומה מזה. אז זה סרטון ב4 הרץ בערך, כאילו זה 4 פריימים בשנייה, שאנחנו מספיקים לקבל איזושהי רזולוציה.
זה שניות כאילו עד מאז שזה מגיע?
כן.
זה באמת מגניב, איך הוא מטייל. זה הליזוזום או אוטופגוזום?
חלק מהם זה גם ליזוזום פעיל שמטייל, כן.
זה שני הכיוונים אתם רואים?
all over the place.
אבל כאילו יש לה רטרוגרייד ואת ה...
כן, אז הטענה הדוגמת, אין פה אנשי אוטופגיה, זה דברים שאתה אומר, בכנס אוטופגיה אנשים מתחלחלים, כי הם כזה חיו על דוגמות איך דברים זזים, אז כאילו הם טוענים שמהאקסון יש רק תנועה רטרוגרייד, שבעצם הם נולדים פנים על הסום, הם נולדים באקסון, והוואקיולות האלה נודדות לתוך גופתה, ובדנדריטים זה all over the place, והם מוסכם, וגם אנחנו גם רואים שבאקסון יש פעם תגיד לאף אחד.
יש לתוך ספיין ונשאר שם?
אז יש הרבה שהם סטטים בתוך ספיין או ליד ספיין, זה מאוד קשה מהסטרקצ'ר.
זה היה עם ויבו?
כן. זה מאוד קשה
וזה עם וירוסים?
זה עם פלזמיד, אבל יש לנו גם וירוסים, כן, גם וגם. אנחנו עושים גם ניוטרו-אלקטרופוריישן, בעובר, ואז עושים אינפובר אימג'ינג, אבל גם עם וירוסים.
אז אפשר כל אנימל מודל.
כן, נכון.
אז ערנו איש
כן, אני יודע, דיברו איתי כמה, אני לא יודעים, אף אחד עוד לא ירים את הכפפיים, כולם אמרו, וואו, מגניב, אולי נעשה את זה.
כבר עכשיו.
כן.
האקסון, מה הוא בעצם מעביר מידע שהוא ראה משהו כאילו?
בית,
בעיקרון הוא נושא חלבונים פגומים או או שומנים פגומים, כאילו זה מה שנקרא מקרו-אוטופג, הוא לא סיגנל ספציפי, יש כל מיני סיגנל ספציפי של
מעביר כאילו יש כדור מהאקסון, מחזיר אותו חזרה.
כן, כאילו הגוף התא אומר הפח זבל, שאליו כל הדברים זזים, ושם יש את המכבור הזה של המסגור.
אבל זה עדיין זה הרבה אנשים.
בטח. כן.